What's up, nerds? Microplastics are everywhere. They're in the water, they're in the food we eat, they're in the children. Oh dear God, not the children, please. If you don't know what microplastics are in the year 2025, you're either living under a bed of them or you secretly are one and you're just trying to blend in so that people don't come after you. But in case you're new, microplastics are these teeny tiny versions that are formed from breaking down the normal big bad versions of plastics that we use in our daily lives. And you might be thinking, bro, the plastics are literally micro. Don't be such a beta. One study showed how many more microplastics were recently found in human brains. Look at how much they've increased in the last 8 years alone. At this rate, our brains are going to be 100% plastic by the year 2046. And there are several reasons why microplastics are everywhere. The detection methods are trash, the removal methods are trash, and the prevention methods are basically non-existent because we as a species love plastic. So do we just give up on trying to remove them and instead accept them as our nutritious and delicious overlords? Hell no. At least that's what this paper out of McMaster University says. A strategy for quantifying microplastic particles in membrane filtration processes using flow cytometry focuses on how we can remove microplastic particles from water and accurately and easily detect every single particle that may have slipped through the cracks. This paper is trying to make a major effort in how we can better detect how many microplastic particles there are. If we can detect all of them, we can learn how to remove them. Buckle up, chemical engineers, environmental scientists, and parents whose children are made entirely of plastic, because this one's for you. Like with all papers, we have to go over some general topics and definitions before we get into the thick of things. Now, after watching the first 30 seconds of this video, I know we're all microplastics experts now, but I really like the formal definition that these researchers give. Microplastics are particles that are often defined as being less than 5 millimeters in size. However, they can be found in the micrometer size range. These particles create a persistent biological hazard in marine ecosystems. Due to their ability to act as both sources and sinks for contaminants, numerous researchers have shown the wide-reaching negative ecological consequences associated with microplastic pollution, with potential implications towards human health, commercial interests, and the economy. Which is exactly what we said earlier. Teeny tiny stinky poison, ew, bad poo-poo yucky plastic, bleh. But where do these plastics come from? The short answer is everywhere. But the best sciencey answer we've got is that wastewater treatment plants produce a lot of it. Wastewater treatment plants are the places that put your stinky poo and or bath water through a series of physical and chemical processes to make it less stinky and eventually good enough to use in your home again. These authors imply that wastewater treatment plants will either form microplastics from their regular bigger plastics as they're going through the plant, or they might simply miss some of them that were formed earlier in the disposal process. Membrane technologies are said to be the ideal solution for preventing microparticles from leaching back into the water that we'll eventually use. Membranes are just filters. These are a solid material that have small enough holes to prevent microplastics and other debris from getting back into the water that we want to use later, but large enough to still allow water to pass through. And usually these membranes are really great at catching plastic particles. For example, this review says that treatment plants can catch anywhere from 70 to 90% of particles. Only around 0.5 to 50 particles per liter avoid treatment plants and eventually get back into the water that will circulate back to our homes. The always accurate Google AI says that an average person creates about 200 liters of wastewater per day. And this source says that a single treatment plant can process 3 to 50 million gallons, aka up to 190 million liters of water per day. So look, I never learned how to do math. When I read these initial stats, I'm thinking how many particles are still in our water even after it's been treated. Even though there's only a small amount of particles per liter that's being missed by these treatment plants, if you consider how much waste we all provide and how many liters of water a single plant can process, there's gotta be like at least 12 particles left over. Terrible jokes aside, it's clear that current membrane technologies used in wastewater treatment plants aren't good enough at catching all of the microparticles that need to stay out of our water. On one hand, it might seem like 50 microscopic particles in an entire large-ish bottle of Coke probably isn't a big deal, but are we sure that it's only 50 particles? That brings us to big problem number 2, which is accurately counting how many microplastic particles are currently in our water. In the introduction of this paper, they list the different methods that are currently being used. Most of you probably don't care what each individual method means for counting these particles, so I've separated them into 2 general categories. On the left are methods that provide a detailed analysis of all of the particles in your sample, but they're all really slow to gather data, they're time consuming, and they're expensive. On the right, we have methods that are fast at detecting and usually need less preparation to run an experiment, but are much less detailed in how many particles are available and what those particles might look like. But the researchers in this paper didn't like any of these methods. Instead, they used something called flow cytometry. I think this is a good diagram of what flow cytometry is. Your sample is put through this little capillary or tube, and every individual particle is put into a single file line. Recess is done, kids. Let's get inside. Some of these particles have a small dye on them, but the others don't. The entire sample then flows through the capillary and a laser shines on it. The laser causes the particles with the dye to shine and the particles without the dye do not shine. There's a little counting process and voila, you can determine how many particles are in this sample, whether they have the dye and whether they don't. Now, this seems weird to me because originally the authors are presenting this idea of a method that doesn't need a lot of sample prep, but if you have to take different water samples and then add different dye to them just to see if plastic particles are there or not, it just seems like a big headache, and in fact, it might even create new issues. But because these researchers are smarter than me, they thought of a way that you could use flow cytometry without having to add a dye. They've even cited how different studies were done that looked at microplastics that did not have a dye on them and how they could give an accurate signal from flow cytometry. Not only that, they could tell you what the microplastic size, shape, and concentration was, so you can get a lot of information without having to do a lot of sample prep. So putting all this together, here's their game plan. 1, take a water sample that's filled with microplastics and put them through a filter or a membrane. 2, use their unique flow cytometry method to see how many microplastics get through this membrane. And 3, compare and ideally improve on the current methods of both filtering and detecting microplastics from water. Let's get into it. It's usually the part of the video where I show all of the data that they've collected and how it relates to the game plan that they set out for us. But I've been specifically asked to stop showing unsexy graphs, so I'll keep it to a minimum for today. That said, I'll never deny y'all the chance to see some really pretty pictures, so here's a look at what the microplastic particles look like for their studies. They use something called field emission scanning electron microscopy to get these incredibly detailed pictures. And without getting too detailed, it's really just a way of using electrons to focus your sample so that you can get a much higher resolution image than those nerds who are just using a regular microscope. They then looked at each and every individual particle in this image and measured their size. They saw that their sizes ranged from 0.5 to 5 micrometers with an average of around 1.4 micrometers. This is approximately 80 to 100 times smaller than the thickness of one of the hairs on your head. Now, these details are very important to remember because filters are designed to cut out specific sizes of particles. For example, most filters that you can buy at your local art store have pores that are a few millimeters in size, so they won't be able to catch anything that's as small as the microplastics that are in the water samples that we see in this research study. That also means that these researchers need a very special type of membrane in order to try and catch as many particles as they can. We'll get back to this later, just hang tight on that. Now that they know what the plastic particles look like, they can design these experiments to see how many are being filtered and if they can detect every single particle that goes through the filter. Not 99%, not 99.99999%, 100% of the particles that are going through. Before they do that, they just need to prove that their specific flow cytometry setup is able to detect particles at all. Fair warning, doesn't really look that exciting. Basically get a bunch of dots on the screen, and you have to decide which side of this picture looks like good dots versus bad dots, or dots you want to analyze versus dots you don't care about. Each red dot in this image is a unique microplastic particle. The authors chose that anything to the right of this black diagonal here is a particle that's worth measuring, while anything to the left is probably just some random background noise. This is important because it proves that microplastic can be detected with their flow cytometry setup without needing to add any of these dyes that we talked about previously. For most researchers, this is a pain, and the more you have to prepare a sample, the less accurate it might seem in the setting that it's going to be used in. For example, no one's going to be adding a dye to millions of liters of water that come through a wastewater treatment plant every day. You want to be able to just test the water without doing anything to it. And these researchers showed that. Hooray for less of a pain! But the cooler experiment is when they mix these microplastics with something called humic acid. This is something that commonly builds up on the membranes of a wastewater treatment plant and prevents them from working properly. Think of it as a contaminant, but it's a very special contaminant because it causes microplastics to aggregate or clump together. And remember, if a filter is meant to have very specific pores, but a particle changes its size, it may interact with that membrane differently. It may or may not pass through. And what they saw was super interesting. Any orange dots in this image is a microplastic particle, but the dark gray dots are humic acid. What we can see on the left side might look a little shocking to you at first. Is that a bunch of orange dots? Why are there so many microplastics over there? And these authors assume that they are in fact aggregates or clumps of microplastics that were caused by the added humic acid. But the real triumph is actually on this right-hand side, where there are clearly still lots of orange dots. It looks almost exactly the same as the first image we saw. So what does this all mean? It means that even in the presence of humic acid, which is something that's designed to screw up how we count microplastics, flow cytometry is still able to accurately count how many of these plastic particles are still available in that water source. Flow cytometry is completely unfazed by the humic acid bowl that is in their microplastic china shop. Importantly, it also gives our researcher pals a whole lot more more confidence to keep doing more experiments with this method without worrying that they're measuring the wrong thing. In general, this is a very important point when doing any kind of scientific research. We have to be 100% confident that the data that we're collecting is actually representing the thing we're trying to measure. This experiment was a very important step, even if it wasn't maybe the flashiest. But on that note, let's get to the flashy stuff. After all this buildup, these authors wanted to answer the question, if we filter these microplastics, how many individual plastic particles can we count using our fancy schmancy flow cytometry method? So they put microplastics either in water by itself or in a solution that contained humic acid. And they passed them through different filters or membranes that had different pore sizes. They then used flow cytometry to determine how many particles were rejected by the filter, i.e. were not allowed to pass through that membrane. Ideally, they were aiming for 100% rejection, meaning that none of the microplastics got through the membrane, and none of the plastics were staying in the water that had previously been processed. If they couldn't catch all of the microplastics, they at least want to be able to count how many got through. Really quickly, let's think back to how big these plastic particles were. On average, they're around 1.4 micrometers big. All right, now turn to your hypothesis brain. Which of these membranes do you think the microplastic particles should be able to pass through? Membranes with pore sizes of 0.45 micrometers or ones with 5 micrometers? Think about it, write it down, we'll come back to it. So like in the previous graphs, the red dots here are when microplastics were in water. We see that only about 95% of plastic particles are rejected or filtered out when using the large 5 micrometer pores, but 100% of the particles are rejected when using the membranes with smaller pores. Was this what you predicted? My guess is yes. When the size of the pores are bigger than the plastic particles can just pass through the membrane and stay in the water sample. When the pores are really small and that plastic is bigger than the pores in the membrane, most of it stays within the membrane, what they calculated to be around 99.5% of the time. But now let's zoom in when microplastics were added to the solution that does the clumping. After only 15 minutes, we see almost 100% rejection in both filters, the ones with the small and the large pores. And this is great. This is realistically what we should expect. Thinking back, if we know that humic acid clumps these microplastics together and makes them much, much bigger, they wouldn't be able to pass through either of the membranes, whether they had big or small pores. What does this mean in the greater scheme of all of these experiments? The authors point out that both efficient and effective microplastic removal performance for particles around 1 micrometer and larger could feasibly be achieved using a 0.5 micrometer pore size in tertiary wastewater filtration processes. These researchers are advocating for the fact that pretty much any wastewater treatment plant can improve their microplastic filtration processes if they just incorporate this really simple, very cost-effective membrane. They don't need anything fancy schmancy. They just need to have pore sizes of 0.45 micrometers, which we wouldn't have known if they hadn't confirmed it with their flow cytometry method. Experiments like these might seem inconsequential at times. Whoop-de-doo, they were able to filter some particles and then count how many they didn't see. But in reality, it confirms how good these methods are. If they can get more people on board with this filtering and analysis technology, more people could remove more microplastics without having to spend an arm and a leg. And if Timu's taught us anything, it's that getting the same performance at a fraction of the price feels amazing. My simple goober brain just thought it was really cool that they were able to detect individual particles of plastic using a method that I didn't even know was possible to use. I thought it was only used in biomedical settings and not in wastewater treatment. And for me, learning nuanced takes is always the most fun part of any scientific paper. But what I think they did best was explain the significance of their work to both a technical and a general audience. You see, most research papers are written for a very technical audience. And this is very helpful because it means that these technical people can come up with brand new technical solutions. But it doesn't really help me, with someone who barely knows how to read, to connect with the paper and the research that you've done. So for example, this whole paragraph here, you don't have to read the whole thing, but like, what in the hell are they even talking about? My scattering, monotonic particle, filtration relationships, bacterial refractive properties. From what I can gather, this paragraph is talking to the technical experts in their field as to why certain things are happening and how they're building on previous data that were provided by other scientists. On the other hand, they have these paragraphs that I think most, if not all people, can relate to the main points they're trying to bring home. In this work, the utility of the flow cytometry technique was demonstrated specifically for quantifying the presence of well-defined microplastic particles suspended in one of two solution conditions as part of a microfiltration process. Given that flow cytometry is already deployed in the biomedical and environmental sciences to count cells of various shapes and sizes, it's feasible that the technique could be extended to systems involving fluorescent microplastics of various sizes and shapes. The technique may be feasibly leveraged in systems with complex solution chemistries, such as municipal wastewater. Ongoing research should be undertaken to rigorously validate flow cytometry in these applications. They're both telling us what worked for them, but how it could work for other others in the future. They're also saying to other people, hey, this was a great start, but we need a lot more work to do in order to get this on a scale that's large enough to be used in municipal wastewater treatment plants and are able to actually help people in their daily lives. Because really that's what science is about, discovering new ways to help people. If you learned something today, like this video and subscribe. If you want to learn more, let me know down below or find the full text link of this paper in my bio. You should also comment over to Instagram to learn about more cool papers, but in bite-sized form. But with that, see you later, nerds.